day, students. Welcome to my virtual classroom. For today's discussion in biochemistry, we will talk about enzymes or the enzymatic catalysis. We will also discuss some properties of enzymes and how enzymes are classified by the type of reaction they catalyze. And also you will know how these enzymes act on a specific substrate and how some enzyme requires cofactors. Living systems are shaped by an enormous variety of biochemical reactions, nearly all of which are mediated by a series of remarkable biological catalysts known as enzyme. And the branch of science that deals with the study of enzyme is called enzymology. Okay, again, enzymology, it is the study of enzymes. Has its roots in the early days of biochemistry. So both disciplines evolved together from 19th century investigations of fermentation and digestion. So initially, the inability to reproduce most biochemical reactions in the laboratory, led by Louis Pasteur and others, to assume that living systems were endowed with a vital force that permitted them to evade the laws of nature governing inanimate matter. Some investigators, however, notably like Liebert, argued that biological processes were caused by the action of chemical substances that were then known as ferments. So indeed, the name enzyme, so yung term na enzyme, en meaning in English, and zyme, or that is yeast, meaning there is something in yeast. Okay, so that was coined in 1878 in an effort to emphasize that there is something in yeast as opposed to the yeast itself that catalyzes, which catalyzes the reactions of fermentation. So eventually, Edward Buchner showed that a cell-free cell extract could in fact carry out the synthesis of ethanol from glucose, and that process is known today as alcoholic fermentation. Okay, next is the enzyme classification according to reaction type. Okay, so we have 6 classification and also their corresponding type of reaction being catalyzed. Okay, first is the oxidoreductases. So the type of reaction catalyzed is the oxidation reduction reactions. Next is the transferases. Okay, the transfer of functional groups. Next is hydrolysis, which is the hydrolysis reactions. Lyases, group elimination to form double bonds. Isomerases, for the type of reaction, isomerization. And the ligases, bond formation coupled with ATP or the adenosine triphosphate hydrolysis. What are enzymes? Describe the structure of enzymes, the types of enzyme reactions, examples of enzymes, and the effect of temperature and pH on enzymes. What are enzymes? So enzymes are found in all living cells. So enzymes are made up of protein. Enzymes are biological catalysts. Enzymes speed up reactions, and enzymes are unchanged by the reaction. Okay. Let us see now the structure of the enzymes. The substrate is the chemical that the enzyme acts on. So can you look now to the image? So the product is the end chemical produced. So the active site of an enzyme is where the chemical reaction occurs, and it is said to be complementary to the substrate. Okay. And the enzyme and the substrate shape match each other exactly. So enzymes are specific as only one enzyme acts on only one substrate. Types of enzyme reactions. Okay, first is the degradation reaction or the breaking down. Okay, just like this one. We have here a hydrogen peroxide. Okay, so we have this enzyme catalase, and this catalase now breaks down this hydrogen peroxide to form oxygen and water. Okay. Next is the synthesis reaction, which is the building up. Okay, we have this glucose 1 phosphate. So using through the enzyme phosphorylase, it builds up and form starch. Next is the examples of enzymes. So we have this following substrate. We have this enzyme and the product. Okay. We have fats. The enzyme would be the lipase. And the product, the end product is the fatty acids and glycerol. Okay. Another substrate is the protein. And the enzyme is protease. Okay. The product is the amino acids. Next is the starch and enzyme amylase. Okay. And the product would be maltose. Okay. Types of enzyme reactions. Okay. A protein molecule is made up of many different amino acids. Okay. And the enzyme protease breaks down protein molecules to form amino acids. Okay. A starch molecule is made up of many glucose molecules. And we have this enzyme, carbohydrase, breaks down carbohydrate molecules to form glucose. Okay. Next, a fat molecule is made up of fatty acid and glycerol molecules. Okay. And it also, these fatty acids now will eventually form into glycerol, and the enzyme that is responsible to break down this is the lipase, and the lipase breaks down fat molecules to form fatty acids and glycerol. Okay. Next, the effect of temperature on enzymes. As temperature increases, the enzyme activity also increases. Okay. So the temperature that the enzymes works best as it is called its optimum temperature. Okay. If the temperature is too high, the enzyme's active site changes its shape. So when the enzyme's active site has changed shape, the enzyme is said to be denatured and it will no longer work. Okay. What are the effects of pH on enzymes? So enzymes work best within a range of pH depending on the type of enzyme. So the pH that the enzymes works best is called its optimum pH. If the pH is too high, the enzyme's active site changes shape. What will happen? Denatured. Okay. So what is the optimum pH of this enzyme? Okay. Based on the figure, makita natin. Optimum pH niya is 7. Okay. Now, what will let us see now the denatured enzymes, or the figure of the photo or the image of the denatured enzyme. See? The normal enzyme active site fits substrate specifically like a lock and key. They are complementary to each other. So, yung active site, yung enzyme, at yung substrate. Okay, lock and key siya. So, fit na fit siya. Okay. And the second picture showed the denatured enzyme, meaning na bago yung shape niya. So, the active site has changed shape. So, it no longer fits substrate specifically like a lock and key. So, active site, enzyme, and the substrate. So, enzymes and industry. So, enzymes in fruit juice. So, enzymes help to get as much juice as possible from the fruit. Enzymes in detergents. So, enzymes help to break down biological stains on clothes. And enzymes in pharmaceutical industries. So, enzymes are used in medicine to treat blood clots and tumors. So, enzymes are also used in testing kits, like for example, testing for sugar in urine. And enzymes and its application to biotechnology. Okay, we have this term immobilization. So, immobilization is a technique used to trap enzymes or yeast cells so that they can be reused in order to save money. So, this means products can be continually made. This process is also called continuous flow processing. Immobilized enzymes are used in the production of lactose-free milk for people who are lactose intolerant. So, immobilized enzymes are used in glucose testing strips to measure glucose levels in diabetics.